Okay, great. I don't know. Can you see? I don't want a big bulk of you guys in here. Oh, come on. Shoot. Okay, break rooms, pause, record meeting info. Well, can you guys see everybody's else name on the screen too? I'm afraid this is being recorded too. Here we go. I got it. I got it. Okay.
I can't
The
see
very
anybody's
first.
name.
Okay. Now you should not. You should see all of the bacteria, nothing else on the screen, right? Oh, is that right?
I just see the slides, nothing written on them.
Great, 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 awesome. So the type of question I will ask for this slide, it would be, what is the name of the organism? This would be a bacteria, okay? And then I might ask what kingdom, it would be kingdom bacteria again. What is the name of the organism as a bacteria? You guys are not learning. I'm not talking about it's a staphylococcus, is a cluster of round bacteria. So what type of cell? I might ask also what type of cell. The type of cell, it is a prokaryote. So type of cell prokaryote, name the organism for now as a bacteria, and what kingdom, it would be bacteria. Same as next slide, okay? These are all bacteria, cluster of them, okay? We will revisit this lab, these slides later on. Okay, this is slide of frog blood. So I see a lot of frog red blood cells, and I don't see frog, here we go. That's a frog white blood cell right there. Everything else, these are all red blood cell, red blood cell, red blood cells, but that's the only white blood cell I see. So the three component, if you refer to your lab manual, the three component of this you have to know. The nucleus, I put a cross on it, that's a nucleus. Then this is cell membrane, and then all around the nucleus right here is cytoplasm. So three things you should know. Okay, the nucleus, cell membrane, right there, and cytoplasm, all of the squiggly lines. Okay, so those are the three components. And of course, you should know belongs to kingdom Animalia, frog blood. That was a quiz question. The white blood cell is right here. You only see one. It might come up later on. Same thing, frog blood. You know, on the lower on scanning power, this is on 10x, and I should have one on high magnification. Frog blood again, that is scanning power. This is 10x, and so this must be 40x. Yeah, these are 40x. So 40x, and that's it. That's, do I have another? Yeah, that's another beautiful white blood cell. Frog white blood cell. Okay, but these are red blood cells. Frog has red blood cells. Have nucleus. We human do not have nucleus. Okay, here is again bacteria. Same thing as I mentioned before. So that's it. That's for bacteria. Same thing again, bacteria. This is cross section of leaf. So this is a leaf. They cut it. They sliced it in a cross section. Okay, so the components that you have to know, these are the opening. You see it in high magnification, which is called stomata. And then this is vascular bundle right here, which is made up of phloem and xylem. You'll see it on high magnification. Again, stomata. This is epidermis. And this layer right here, not this layer and not that layer is called endodermis. Okay, epidermis, endodermis, vascular bundle. These are the things. And I think in the photosynthesis lab, I mentioned all of these, okay? This is in the photosynthesis lab. Okay, this is a high magnification. You can see beautifully the stomata, the opening. And then there are guard cells here. I don't think I mentioned the guard cells in your lab manual. So don't worry about it. This is vascular bundle, the whole entire thing. And the red is xylem, and the green, all of the green right here is phloem. So xylem is the red, and the green is phloem. When you take botany, you will hear more, you will see more. Here is stomata, the opening, guard cell, I don't think I mentioned that. Epidermis, all of this is epidermis. Can you guys see this while I'm doing what I'm doing? Yeah, Ryan?
Yeah, I can see and hear.
Very good, very good, very good. Okay, and then these little dots, you guys see these little dots? Those little dots are chloroplast. These little dots right here are chloroplast. And then of course, this is the endodermis right here. Xylem and phloem. Okay, so xylem and phloem. Make sure you look up those, how to spell those words in your in your lab manual, in your handout. Okay, this is the bread mold. Common name is bread mold. Scientific name is Rhizopus. So what happens, these little sacs, they have full of spores. I'm drawing, you can see it. And what happens, these sacs breaks and little spores get into the air, and they go find another piece of bread and mold that bread. So that the name of the organism is Rhizopus. The name of the sac right here is called sporangium. This is called zygospore. Again, same thing, bread mold. But these are zygospore. For example, this is negative, this is positive. They come together. They don't call it male and female. I don't know why. They call it negative, positive. Maybe nowadays they changed it to, I don't know, male, female. But they call it negative, positive, and they come together and they form the zygospore. Right. Okay. And these are zygospore again, and sporangium. This is called stalk. Sporangium, they are full of spores. When it breaks, it gets into the air. And okay, this is the unfertilized egg of starfish. The reason I say it's unfertilized, because you can see the nucleus. Right here is a nucleus. And inside of the nucleus is a nucleolus. This is nucleolus. And then of course you have cytoplasm, you have cell membrane, and then you have nucleus, of course, and then inside you have nucleolus. Okay, so that's a starfish unfertilized egg. You can see the nucleus. Okay, this is Spirogyra. It's an aquatic plant, and all of these are chloroplasts. You see these wiggly lines in here, kind of, sort of, right here. They're all chloroplasts. And then I do not have a slide that it shows you the nucleus and everything else in there, but they have nucleus, and these are plants, aquatic algae. Here again, the starfish egg, which is not fertilized. Right here is the egg, right here is the nucleolus, and right here is the cell membrane, and right here is the cytoplasm. Okay. Again, same thing again. Starfish unfertilized egg. This is starfish fertilized egg. You cannot see the nucleus. You can see a little bit of a nucleus of sperm and egg. So this is the fertilized egg, or zygote, if you would. So that's one. This is another one, another zygote, fertilized egg of starfish. Okay, this is paramecium right here. These are paramecium and belong to phylum to kingdom Protista. The type of cell is they are eukaryotic. They are nucleus, of course, right here, and cytoplasm right here, and they have cilia. They move by means of cilia, high magnification, nucleus, cytoplasm, cilia, all around it. Okay, I hope I'm making some sense. Eukaryotic cell, kingdom Protista, what else? What else? Whatever else I listed in your lab manual. Amoeba. So in the amoeba, these are pseudopod. They have pseudopods, pseudopod, and then this is the nucleus, and this is the vacuole right here. Okay, kingdom Protista type of cell is eukaryotic. I hope I'm straight. Okay, onion root tip on low magnification, not a very good one. I probably will not put it in the exam, but here it is, a much, much better one, onion root tip. And these are nucleus. Inside of them, you can see the nucleolus. Some of them, its nucleolus is perfect. These are chromosomes right here. And they're dividing. That's the next exam material. But you should know the 3 components or 4 or 5 components in here: cytoplasm, nucleus, nucleolus, cell wall. And then, of course, chromosomes. So 1, 2, 3, 4, 5. So 5 things you should know in this slide. And then, of course, is onion root tip, belongs to kingdom plant. It's eukaryotic cell. Same thing again. These are the nucleus of the onion root tip. And these are, these are all nucleus. Okay, let's go. Same thing again. Later on, you will learn another component into these slides, which is right here. It's called cell plate. But right now, do not worry about it. This is chromosome. This is chromosome. These are chromosome. These are chromosome. These are nucleus. And you're going to learn some more next exam. Okay, unfertilized egg. Fertilized egg of starfish. Fertilized egg of starfish. Unfertilized egg of starfish. This is Tilia stem. So it's a plant, kingdom plant, TA. Eukaryotic cell. This is the stem again, the longitudinal cut. The last one was a cross-section cut. This is a longitudinal cut, a cross-section of a different plant, but let's call it Tilia. I think I gave the name in your lab manual, but both of them are Tilia. Again, I'm a little bit fuzzy on which one is Phyllis and which one is Tilia, Phyllis, and which one is Tilia. But anyhow, okay, bacteria. These are the spirillum, the wiggly bacteria. Okay, so again, they're prokaryotic kingdom bacteria, and so on and so forth. This is Trichomonas vaginalis. You can see they have flagella in them. Okay, there are 4. I think there are 4 flagella. You can see the nucleus in some of them right here. Some of them are big enough. Right here, you can see nucleus. So phylum, this is the one that causes trichomoniasis in female. It's sexually transmitted disease. And in female and male cause trichomoniasis is more common in female than male. But anyhow, that's a different story. I will talk about it when you get to zoology. What is? Kingdom Protista, eukaryotic cells, mode of locomotion by flagella. And then, of course, you have cytoplasm here, a little bit deeper. Yes, cell membrane here and flagella here. So 4 or 5 components here you should know. And then, of course, eukaryotic. Same thing again, Trichomonas vaginalis. Trichomonas vaginalis on a lower magnification, a scanning power on the microscope. Here is a photosynthesis, which I did talk about it in the YouTube video. And then I don't think I have anything else to say. Yes, I am ready for questions. Any of you have any questions? Anybody, any question? Thank you, Ryan, for bringing that up. I'm being recorded, so if you guys do not have any question, please let me stop recording. But we still, it doesn't mean we end the session. We will not end the session But if you do not have any question, let's pause for a few seconds, and then if you don't want to be recorded, then I can stop recording. Yes, go ahead, Ryan, please.
Well, I didn't have any questions. I was just saying that that's what I really wanted to do, was go over the slides.
Okay, so let me stop recording.